I'd like to welcome you to this webinar about automatic annotation systems in UniProt. The content will be delivered by Alex Renaud and myself, Clemens Pichler. We'll swap over in the middle of the talk. And in order to start talking about automatic annotation systems, we first have to set the stage a little bit by talking about what UniProt actually is. UniProt aims to provide the highest quality, most comprehensive, and most thoroughly annotated protein resource. As such, we include detailed information about protein function, interactions, pathways, all aspects of a protein, really. We also obviously provide the sequences, including any isoforms of a protein, known disease variants, and post-translational modifications. Last but not least, we also provide stable identifiers. We usually call them accessions, and we urge people to use those to refer to UniProt entries and sequences in their publication because they are stable and are traceable. Obviously, providing UniProt resources is a major endeavor. Many people are part of this. They're all listed here. I'm not going to go over them one by one. And also, we couldn't do this without the financial support of many funding agencies, which are also listed here. Resources that UniProt provide are manifold, at the center of which is the UniProt Knowledgebase, here in the blue box in the middle. And this is also what most people refer to when they talk about UniProt. But there are other data sets as well. For example, a specific view on proteomes. Then there is a UniRef, a resource on sequence clusters for sequence similarity searches. And there is, underpinning it all, the UniProt archive that contains all the sequences in the public domain and many more than you would find in the UniProt Knowledgebase itself. But today we'll talk about the UniProt Knowledgebase. The UniProt Knowledgebase consists of two data sets, one of which, also known as SwissProt or the reviewed dataset, is only about 500,000 sequences or entries strong. On the other hand, there is TrEMBL, or the not reviewed automatically annotated dataset, which is huge by comparison, with currently about 100 million entries in the public release. However, SwissProt, the smaller dataset, contains all the expert curated information on all aspects of a protein, for example, its function, or any residue-specific data that we can find, and these can be active sites, for example, or disulfide bonds. SwissProt also contains protein isoforms pertaining to one gene in one UniProt Knowledgebase record per species, obviously. This is a different concept from TrEMBL, where protein isoforms of one gene would be found in different UniProt Knowledgebase records per species. That is because the computers do not make a call as to whether sequences that differ in even just one amino acid belong to the same gene or not. This is left to specialized personnel, the curators that work on SwissProt. TrEMBL contains a lot of information despite being automatically generated, for example, mapped experimental sequence features from 3D protein structures, and also all the annotations from rule systems, including expert curated rules, feed into TrEMBL, and this is what automatic annotation is about. So how are the two datasets connected? Starting from the top here in this flowchart, the unreviewed TrEMBL part of the UniProt Knowledgebase, this is where sequences are selected by people, biologists. Then they undergo a process that we call expert curation before they're integrated and end up in the reviewed, the SwissProt section of the knowledge base. As part of the expert curation, what kinds of data are added and collated for each individual entry that we generate? Obviously, being a protein sequence database, we have sequences pertaining to each entry. That includes, I've mentioned it before, protein isoforms, variants, and all different kinds of sequences that belong to the same gene. We also collate all the functional annotation that we can find about a given protein. That can be subcellular location, tissue specificity, expression in a particular developmental stage, catalytic activities, and the like. If functional annotations can be tied to specific amino acid residues, we also capture this. And this is something that I think no other database, even protein-specific database, does. We collect all the names that given proteins are referred to in the literature, providing a comprehensive collection so that whichever name you use for searching, you can hopefully find the protein you're interested in. All the while, we try and enforce a standardized nomenclature for proteins as far as possible. I've mentioned the identifiers, the accession numbers, which are unique and stable and should be used in publications. We also link out to a great number of specialized databases, and this is something that I'd like to come back to later on. In addition to providing gene ontology terms, and also we care a lot about taxonomy, because we'd like to make sure that information ends up in an entry of a given species and not in a wrong one. All of these boxes are filled primarily by taking data from the scientific literature. We also use bioinformatics predictions. This is mostly used for signal peptides or transmembrane domains, for example. We collate all the sequences. This is the part of the sequence curation that's mentioned in the gray boxes here. And within protein families, we try and standardize the available information and its presentation as much as possible. This is also something that feeds into automatic annotation. Entries undergo several stages of quality control before they're made available publicly, and part of this quality control is something called evidence attribution, which is also very important and linked to automatic annotation later on. So what is evidence attribution? An evidence basically describes the source of an annotation, where an annotation is any piece of information contained in a UniProt Knowledgebase entry. These evidences are based on a structured system called the evidence ontology, in case you'd like to read up on it. How does it work? Say we have a particular protein X where we assert that this is an alcohol dehydrogenase, so an enzyme. A curator would then go off and find all the sources that provide the data that support this assertion, basically reading the papers that describe the biochemical experiments proving that protein X indeed is an alcohol dehydrogenase. Once this has been done, all the information and the necessary steps is encoded in a little label which is then attached to the annotation. And this label we call an evidence tag. There are many different evidence tags, and the important thing in relation to automatic annotation is that they can be used for filtering searches when running searches on the uniprot.org website. Here is a screenshot of the advanced query interface on uniprot.org. In this instance, targeting the function free text comment contained within entries. And here, different evidence levels can be chosen. And the one that are most relevant to automatic annotation are boxed with a black dotted box. Now we can move on to the automatic annotation systems. Coming back to the initial flowchart that I've shown before, where data is taken from the unreviewed part of the UniProt Knowledgebase and through expert curation feeds into the reviewed SwissProt part, there are challenges here which I've failed to mention so far. First of all, the unreviewed part, the blue bit at the top, grows very fast. I've mentioned at the beginning that it's about 100 million entries currently, and this is set to grow and will keep on growing. The expert curation, reading papers, running bioinformatics analyses, etc., takes time. But even if we had more personnel and could have more people working on this, the fundamental problem with annotating protein sequences is that for many of them, there is simply no experimental data available. So the only way to annotate them is by drawing on experimentally characterized sequences and annotating the ones without experimental data by similarity. And this is basically what automatic annotation is about. On the just revealed right-hand side of this flowchart is the automatic annotation pipeline part of the data flow within the UniProt Knowledgebase. And this time it starts at the bottom, drawing on all the effort that has gone into the reviewed part of the UniProt Knowledgebase, extracting and distilling knowledge and information, encoding them in a formalized way, which we call rules, and feeding that rule output back into the top bit, the TrEMBL unreviewed part of the database, making it immediately more useful. So how do rules work? Imagine for a second that this little box at the bottom of the slide with the colored shapes represents the unreviewed sequences in the UniProt knowledge base. We could now define a few conditions that, if met, trigger some annotations to be applied. These conditions in this example could be that we select all the shapes that are of color purple and have 4 edges. This would give us a subset of all the available sequences. We could then apply an annotation that says all of these selected sequences are actually purple quadrilaterals. So this way we could describe that subset of sequences and make it more useful to the user who wouldn't know anything about shapes beforehand. We can go one step further and add another set of conditions and annotations on top, saying that out of those initial sequences that we've selected or shapes that we've selected, if their edges are of the same length and the angles are 90 degrees, let's call them squares. So you see that we can have nested structures where we provide ever more and ever more detailed information about given protein sequences and adding it to make it available to the users. What are the real-life conditions that we use? Because obviously this is not about geometric shapes and colors. What we use primarily are InterPro signatures. More on this on the next slide. And in addition, taxonomy. There are further condition types that we can use. For example, one can implement length restrictions. And there are some particular features attached to proteomes. However, both of these are of very limited use and pale in comparison to the importance of the first two, the InterPro signatures and taxonomy. InterPro basically allows functional classification of proteins using predictive models, which we refer to as signatures. InterPro, on the right-hand side, acts as an umbrella vessel collecting all the efforts of many different research groups shown in the middle. That's the blue shape, the colored shapes, that try and describe different biological entities using different methods. Both the biological entities and the methods are shown in gray on the left-hand side. Without going into too much detail, what's of most interest to the automatic annotation systems are the biological entities that are described by the resources and are boxed with a dashed box in the middle. So those are domains and families and features and sites. How does this connect back to the data that we have available in the UniProt knowledge base? We've had this slide before where we saw all the different types of data contained in the UniProt knowledge base and in its entries. One of those data boxes was the links to specialized databases. And it turns out that all the member databases that InterPro collects from, that we've just seen on the previous slide, are actually linked out to from all the UniProt knowledge base entries that have a connection to one or many of these. In addition, I've already mentioned that we care a lot about taxonomy and make sure that information is always attached to the right kind of species and its protein sequences. So both these very important condition spaces that we need for automatic annotation are already taken care of by the information that's readily available in the UniProt knowledge base entries, both the reviewed ones and the unreviewed ones, I'd like to add. In terms of annotations, the rule systems that we use, and the first one I'm going to talk about is one where actually people create the rules for them to be applied automatically onto the unreviewed part of the database. Those are not restricted in any way in their ability to propagate annotations. So they can provide functional annotation, amino acid specific data types. They provide names for protein sequences and propagate gene ontology terms. There's only a select few restrictions where the data are so species specific that they can't be propagated with any certainty to other species, for example, tissue specificity or expression within developmental stages. Those are excluded from automatic annotation predictions, but all the rest is fair game. With that said, here is a real-world example of a rule that was is currently being applied within the automatic annotation pipelines, within the system that we call UniRule, that is run by people, where people create the rules. As you can see on the left-hand side, there are a very few conditions which, if satisfied, trigger the annotations on the right-hand side. In this instance, there is one InterPro signature, in particular a PFAM signature, that's one of the member databases of InterPro, and a taxon restriction, which means this is only applied to fungal sequences. If these conditions are met, then the corresponding sequences get a name, which in this instance is lysophospholipase. They also are annotated with a predicted catalytic activity and are put in a particular family, the lysophospholipase family of protein sequences. So imagine that you came across a sequence in a blast search where the provided name would be uncharacterized sequence, but after application of this rule, it would become a lysophospholipase. So immediately it would be more useful to you, and you're more likely to be able to use it in your research going forward if you know what you're dealing with. How would you find such a rule on the UniProt website? In the middle here is a screenshot of the query interface, the query box on the uniprot.org website. And with this little arrow on the left-hand side, you can select different data sources to search. And one of them is UniRule, the system I've just talked about, and SAAS, another system which my colleague is going to talk about in a minute. And that way, if you search that space, for example, for lysophospholipase, you would find the rule that we've just talked about. With that said, I'd like to once more come back to this flowchart, where down at the bottom right, you see that there are automatic rule systems, which my colleague Alex is going to talk to you about now.
So, as Clement said, I'm going to talk about SAAS, which is our fully automatic annotation system at UniProt. So SAAS stands for Statistical Automatic Annotation System, and it is a machine learning algorithm that is using the expertly curated entries from TrEMBL to learn and generate models in the form of rules, which are then applied to the unreviewed protein in TrEMBL to generate automatically annotated proteins. SAAS is learning through a decision tree classifier using the C45 algorithm, and it learns on different kind of attributes, including the InterPro signatures, the taxonomy of the organism of the proteins, and the sequence length. And the learning result of this algorithm is in the form of a tree, which annotation rules can be extracted from, and the rules that are created can be applied then on the TrEMBL entries. To explain how the decision tree learning works, here is a simplified example where we imagine that SwissProt is only made of 5 proteins. We know whether or not it has certain attributes. So here, InterPro signature, a taxonomy, and a PFAM. And we know also the target annotation, which here is transcatalase, which can be present or absent. So here's how the algorithm works. So at the first step, by using these InterPro attributes, which is the one that is separating, that is discriminating the two sets into proteins that have these attributes and that proteins that doesn't have this attribute. You can see already that this set doesn't have this annotation transcatalase. So for this set, you need to discriminate against using another attribute, and the algorithm will select another attribute, which is here the PFAM, to split this set into two different sets of proteins. So the one that has these PFAM signatures on top of the InterPro signature, and another set which doesn't have this PFAM signature. And this last set, as you can see, by having those conditions, has this transcalotase annotation. So by using this algorithm, you can see easily that you can create a rule that will say that if this InterPro signature is present and this PFAM signature is also present, then you can infer the transcalotase annotation. So this rule here in this example has a support of 2, which means that there are 2 Swiss prods supporting this rule. But obviously in our production system, we need a higher support, and we use in general more than 50 Swiss prod entries to consider a rule valid. We also have other criteria like the confidence that has to be higher than 95%. And the rule has to use at least 1 taxonomy criteria and 2 InterPro signatures. So this example is just to simplify, but in reality, it's a bit more complex than that. So it is possible as well as for UniRule to explore the SAAS rules in the UniProt website, either from the main page, as Clement showed you, but also from an entry. If you click on an entry that has been annotated by SAAS and clicking on the SAAS annotation evidence, you can access the rule. And from this rule, you can actually also see all the entries that have been annotated with this rule. And as you can see, as a difference to the UniRule systems, here the rules are much simpler with some conditions, but you don't have those nested conditions. And every one of those rules only annotates one annotation. UniRule was a bit more exhaustive. For SAAS, it generates a simpler rule. And now I'm going to talk about our future plan for our 2 rule systems. So we plan to share the UniProt annotation rules, so for UniRule and SAAS and maybe other systems in the future, and we want to share those rules publicly. The goal would be to open the UniProt systems to the community, but also to facilitate the exchange of rules between the different research groups around the world. We want to provide a way to apply those rules on your own data, and it can be done through a standalone tool or through a web service. And we would like this project to be open to contribution. That's why we need your feedback, and that we want to make it open source and open to improvement. So at the moment, we are designing on one end an exchange format for the rules that will be common for our rule systems and easily processable. And on the other hand, we are developing an engine to apply the rules on your own sequence and retrieve the corresponding annotation from it. At the end, the idea would be to give your protein sequence and as well as the corresponding taxonomy data and the InterPro scan results. So InterPro scan is the tool from InterPro that allows you to run to detect those protein signatures, so InterPro signatures from your own sequence. And by injecting all those data into a rule engine and also injecting either all the UniRules or all the SAAS rules that we will provide, you will be able to infer some protein annotation that will be predicted on your own sequence. So if you are interested by this project, actually, so if you would like, for example, to annotate your own data, your own sequence with the UniProt rules. If you have your own bioinformatics pipeline that you would like to enrich with UniRule and SAAS systems, you can register your interest. There is a link here on the UniProt blog that will give you access to a survey where you can register your interest, and we will be in touch with you. Or you can also contact us directly using the help@uniprot.org email address. Yeah, so that's it for me. Clemens will give you then an overview on how you can access the UniProt data.
Okay, just to wrap up, you might already know if you've used UniProt before that we release new data every 4 weeks. So in between, the data are stable, and all of this is freely available. Obviously, there is much more to UniProt Knowledgebase in particular and the data types it contains than we could have covered in this short webinar. So the second bullet point on this slide points to online training that allows you to explore more about the UniProt Knowledgebase and the functional data that we provide. There's also another link to online training covering InterPro, since this is so instrumental to automatic annotation. And if there are any questions at any time about UniProt related issues, questions, data, download formats, etc., you can always contact our help desk using help@uniprot.org. So with that said, we're at the end of our webinar. We'll stick around for a little bit longer in case there are any questions in the chat. If you did not manage to catch the start of it, for example, there will be a recording of this webinar put up on the EBI training pages. The materials are available in a download tab in your materials section if you want to get them now. This webinar will be recorded and made available for later use if you'd like to come back to it, for example. Thank you for your interest.